Similarly to hairs, spines, and feathers, reptilian scales also develop from placodes, which are small, localized developmental units that act as signaling centers on the embryonic skin. The spatial organization of placodes is established through a Turing reaction diffusion process that can lead to a somewhat irregular pattern, as observed in this mouse embryo. On the other hand, snake embryos develop a strikingly ordered array of placodes that produces a near-perfect hexagonal pattern of scales on the back and flanks, whereas the ventral scales form as a single row along the animal's body. In our paper published today in Science Advances, we demonstrate that ventral scales develop first and are perfectly aligned with the position of somites, which are paired blocks of mesoderm that form along the animal's body and will give rise to many segmented structures such as the vertebrae, ribs, and skeletal muscles, as well as the dermis of the skin. We further strengthened our hypothesis of a tight link between the development of ventral scales and the presence of somatic structures by showing that skeletal defects, for example an additional rib, also cause the development of an additional ventral scale nearby. We then discovered that the development of these ventral scales, under the control of somatic structures, does not require the expression of the canonical EDA pathway. This is highly surprising because this pathway is essential for proper development of skin appendages in birds, reptiles, and mammals. For example, as previously shown in our laboratory, lizards with a mutated EDA gene do not develop scales. We then produced the first genetically modified corn snakes using CRISPR-Cas9 to disrupt the EDAR-AD gene. The resulting offspring lack dorsolateral scales but exhibit ventral scales, unambiguously proving that ventral scales are not self-organized and develop without a functional canonical EDA pathway. The second major result of our study pertains to the development of the dorsolateral scales. We show that once the pattern of ventral scales is established, rows of lateral placodes start to form. This row of placodes is guided by the position of the ventral scales, with which it is positioned in antiphase. Then, additional rows of placodes successively appear as a wave travelling towards the vertebral column. As such, the new placodes form an antiphase with those of the line before, ensuring precise ordering of the pattern. At the same time, a second wave of expression appears next to the dorsal midline and travels both ventrally and dorsally, forming again successive rows of placodes. The dorsal waves are also guided by somatic structures. The two waves meet laterally to form a near-perfect hexagonal pattern. Defects in their alignment are very rare and occur when the two waves meet. Our numerical simulations, based on a reaction diffusion system, allowed us to decipher the self-organizational process that generates the dynamics described above. In the absence of traveling waves, many defects, indicated in green and red, appear in the topology of the scale lattice. When two opposing traveling waves are implemented, far fewer defects appear. When the initial rows are pre-patterned by the positions of the ventral scales and the unidentified dorsal somatic structures, a minimum number of defects appear, as observed in real snakes. These computer simulations indicate that both the presence of developmental waves and their initial pre-patterning by somatic structures are required to reproduce the near-perfect ordered hexagonal pattern of scales seen in real snakes. Remarkably, our analyses indicate that the intrinsic length scale of the self-organized patterning system of snake scales has been tuned during evolution to match the periodicity of the somites. We reason that this match ensures the alignment of muscles, ribs, and scales, all of which play critical roles in snake locomotion. To sum up, our study yields three major conclusions. First, ventral somatic structures induce the development of ventral scales through a yet undefined mechanical and/or molecular signaling mechanism. Second, the first gene-edited snakes produced here demonstrate that the development of ventral scales does not require a functional canonical EDA pathway. Third, the position of the ventral scales acts as a pre-patterned initiation of the dorsolateral placode developmental wave, leading to a near-perfect hexagonal pattern of scales.